Dr. Lauren Rice, who's from the University of Sydney. I had the pleasure of watching Dr. Rice complete her PhD. We can no longer call her an up-and-coming researcher. She's well and truly established at Kids Neuroscience, University of Sydney. Thank you.
My name is Lauren. I'd like to thank Kathleen and Diane for inviting me to present today. I'm going to talk about a study that we've just had accepted for publication. So it's looking at the autonomic nervous system, which Professor Tony Holland has spoken about this morning. So essentially, the autonomic nervous system is made up of two main parts. The sympathetic nervous system is activated when we detect a threat in our environment and promotes our fight and flight response. And the parasympathetic nervous system is activated when we feel safe, and it allows us to feel calm and engage with our environment, including allowing us to socialize. So there's growing evidence to suggest that the autonomic nervous system is altered in people with Prader-Willi, which is throwing off the balance between these two systems, making it difficult for people to stay calm and causing them to be in a state of hypervigilance, where they're overly aware of potential threats in their environment. And this can be a threat to safety, but it can also just be a threat to something they find rewarding, such as food. So approximately 75% of the parasympathetic nervous system is controlled through our vagus nerve, which is the 10th cranial nerve that carries information both to and from our brains to our hearts, our lungs, our stomachs, and other vital organs. And as Tony kindly spoke about this morning, he and his team conducted two small but very exciting clinical trials that showed that vagal nerve stimulation can help reduce temper outbursts for some people with Prader-Willi syndrome. And these have led to a larger clinical trial that I understand Dr. Deepan is leading. So our team were interested in understanding how this device is working and whether there are other characteristics associated with Prader-Willi that might be involved in vagal nerve activity. And vagal nerve activity is known as vagal tone. It's how well our vagal nerve allows us to stay in that state of calmness. And it can be measured using respiratory sinus arrhythmia, which is a combination of high-frequency heart rate variability and respiration. So vagal tone, or RSA, is known to be lower in males than females in the typically developing population and in people with psychosis. So we were interested in understanding whether these trends are also present for people with PWS, and also whether there are differences in vagal tone across the genetic subtypes. We were also interested in looking at whether there are any other behaviours that might be associated with vagal tone that we know play a role, that the parasympathetic nervous system plays a role in. And then lastly, we were interested in looking at whether vagal tone, oxytocin and vasopressin had a relationship, as we know they interact with each other in the brain and that they're all altered in people with Prader-Willi syndrome. And in a previous study that we published, we found some differences across genetic subtypes, so we looked at this separately for those with deletion and UPD. So because vagal tone, or RSA, is known to fluctuate during childhood and adolescence, we recruited 30 adults with Prader-Willi syndrome and 30 age-matched controls. And our cohort included 15 people with UPD, 11 with deletion, and 4 with either unknown genetic subtype or of the rarer types. So we used ECG to measure RSA, low frequency heart rate, and heart period, though I'm just going to talk about RSA today just due to time. And then the behaviours that we were interested in included temper outbursts, skin picking, repetitive behaviours, obsession with an idea or activity, difficulty dealing with change, hyperphagia, daytime sleepiness, and one's ability to recognise emotions in facial expressions. And then we measured vasopressin and oxytocin in plasma, as well as oxytocin in saliva levels. So we found that RSA was lower in people with Prader-Willi syndrome compared to age-matched controls, and this is consistent with the only other study that's measured RSA in Prader-Willi. However, when we stratified by genetic subtype, we found that RSA was significantly lower in people with UPD compared to those with deletion, and also compared to the typically developing controls, but that there wasn't a significant difference between those with deletion and the controls. RSA was also lower in people with Prader-Willi syndrome who had psychosis compared to those who didn't. And initially we thought that the difference in genetic subtype might be because those with UPD are more likely to have psychosis. However, in our study, of the 5 people who had psychosis, 3 had UPD, but 2 actually had deletion. There are a range of other factors that can influence heart rate variability in RSA, and we were able to control for age, BMI, IQ, and use of antipsychotic medication, and these findings remained. We also looked at whether antidepressants might influence the results, and there was no difference in the cardiac indices. In regards to sex, as expected, RSA was lower for males and females for the control group, but interestingly, this sex difference wasn't present for those with PWS. And in regards to the types of behaviours associated with RSA, we found lower RSA was associated with higher rates of skin picking and temper outbursts. And this is consistent with the 2 small clinical trials that suggest that regulating the vagus nerve with vagal nerve stimulation can help improve temper outbursts. As I understand it, skin picking wasn't found to improve in those 2 small trials. However, they weren't the focus, and I believe the larger clinical trial that's currently underway in the US has included a measure of skin picking, so it'll be interesting to see what they find. And then lastly, we found that RSA was, so lower rates of RSA were associated with lower rates of vasopressin, but only for the participants with UPD. And in our previous study, we found that vasopressin was lower for people with PWS compared to controls for both genetic subtypes, but that low RSA was only associated with high behaviour problems for those with UPD. So overall, our findings suggest there is autonomic nervous system dysfunction in Prader-Willi syndrome that may be more marked for those with UPD than deletion, and may be due to a greater loss of parasympathetic activity. So that ability to be able to put a brake on our sympathetic nervous system and allow us to stay calm and engage with our environment. Our findings suggest that future clinical trials should see whether people with UPD respond differently to treatments that target the autonomic nervous system. And interestingly, the 2 clinical trials of vagal nerve stimulation only included participants with deletion. So I don't think we yet know whether vagal nerve stimulation will work for people with UPD, but our findings suggest that it might work better. And lastly, our findings suggest that RSA might serve as an early biomarker for temper outbursts and skin picking in future clinical trials. And that could be clinical trials of vagal nerve stimulation, but it could also be pharmaceutical products or psychological interventions that target the vagal tone. That's it. Thank you. Oh.
Does anyone have any questions for Lauren? An outstanding study, thanks, Lauren.